DNA methylation is the mechanism behind an interesting phenomenon that's known as genomic imprinting. In a nutshell, genomic imprinting refers to the methylation patterns that are specific to the egg and the sperm. When we think about the alleles we inherit from mom and dad, we may think that these alleles have an equal chance of being expressed, and for most genes that is true. This is why if you're heterozygous for a particular gene and one of your alleles is associated with disease, usually the normal allele can compensate for the missing or defective copy. However, there are some genes for which that is not true. There are approximately 100 mammalian genes for which the paternal and maternal copies are not equivalently expressed. One of these two alleles is off by default, while the other allele is on. That means that you get some genes from your mom and some genes from your dad that will never be expressed. Now, although the DNA, the chromosomes are still there, that DNA will not be transcribed into RNA in any cell. And that's the result of genomic imprinting. So it refers to the methylation patterns that are applied during oogenesis and spermatogenesis. Now, this means that in women, some regions of the DNA in the oocyte, which is haploid, will be methylated, but these regions will not be methylated in the sperm. The sperm will show a different pattern of methylation complementary to the eggs. Let's use an example to illustrate this, and this is actually a very complex example because it involves the methylation of an insulator. We're looking at the region of the DNA where you have two genes, H19 and IGF2. We're going to focus first on what happens with IGF2. Now, IGF2, or insulin-like growth factor 2, is a gene that promotes cell growth and also cell division, and it's expressed early in development. Now, part of the figure, part A, shows the maternal copy of the region that contains IGF2 and H19, while part B shows a sperm-derived or paternal copy. The maternal allele lacks a methyl group in this insulator, DMR, which is present in the paternal allele. Because of the lack of the methyl group, this insulator factor can bind to the insulator sequence, and the presence of the factor blocks the enhancer from being used for activating the transcription of IGF2. So IGF2 is not expressed from the maternal allele due to the presence of the insulator factor. On the other hand, H19, which this enhancer also activates, is expressed, since the insulator factor has no effect on the interaction of the enhancer and the H19 promoter. So in the maternal copy, IGF2 is off by default, whereas H19 is on by default. In the paternally derived allele, there is a methyl group that's present at the insulator, so that prevents the insulator factor from binding to the insulator sequence. As a result, the enhancer in this region can activate the IGF2 promoter. So IGF2 is expressed from the paternal copy. At the same time, this methyl group represses the expression of H19. That's why I mentioned earlier that when we're talking about DNA methylation, for enhancers and promoters, it is repressive. But as you can see here, if we add a methyl group to an insulator, it has the potential to lead to the activation of certain genes. What happens when there are defects in the methylation patterns of imprinted genes? Well, if a methyl group is added to the maternal allele during oogenesis, then the maternal allele will look exactly like the paternal allele. So that means that the baby is going to show biallelic expression of IGF2. So both copies of the IGF2 gene will be expressed, and there will be no expression of H19. So in other words, H19 will be off and IGF2 will be on, but we'll have twice as much expression of IGF2 as normal. This condition exists in humans and is known as Beckwith-Whitman Syndrome. Now, since IGF2 promotes cell growth and cell division, children who have BWS show prenatal and postnatal overgrowth, so you can see. So the child will have an enlarged tongue, and they also have predisposition to develop certain types of embryonal tumors. Now, since IGF-2 expression is important early on, but decreases as the child ages, adults who have this condition can generally lead normal lives. A paternal imprinting defect can occur if a methyl group is not added to the insulator during spermatogenesis. So in this case, the paternal allele will look exactly like the maternal allele. Now, the result of this is that there will be no IGF-2 made and biallelic expression of H19. Now, since IGF-2 is important for cell growth and for cell division during development, the child is smaller than normal as a result of prenatal and postnatal growth retardation. This condition is known as Silver-Russell syndrome and is associated with increased risk of perinatal mortality as a result of the growth retardation. As you can see, children who have this syndrome also show distinct facial features. Additionally, although their heads are actually the normal size, their bodies are very small. They're smaller than usual. So their heads appear larger compared to their bodies. In addition to that, they may also experience cognitive delays. Now, an important thing I should note here is that Silver-Russell syndrome does not necessarily have to result from an imprinting defect. What I mean by that is if there is a mutation in the IGF-2 gene, specifically in the paternally derived copy, then that IGF-2 gene, if the mutation is an inactivating mutation, no IGF-2 will be made from the paternal allele, in addition to the fact that no IGF-2 is being made from the maternal allele by default. But if the mutation in the gene was actually in the maternal copy rather than in the paternal copy, then a mutation in this maternal copy would have no effect because that gene is already off by default. Because of genomic imprinting, for some genetic disorders, the expression of the phenotype depends on whether the mutated allele is inherited from the mother or from the father. The classic example of conditions in which genomic imprinting plays a part are Prater-Willi syndrome, or PWS, and Angelman syndrome, or AS. Now, both of these conditions are caused by deletions or other mutations in the same region of chromosome 15. However, part of this region is imprinted or inactivated on the maternally inherited chromosome. So the PWS region, you can see it's inactivated on the maternal allele, whereas the AS region is active in the maternal allele, which is represented in the purple, and inactive in the paternal allele as represented here in the blue. If, for example, this region of chromosome 15 is deleted or inactivated in the paternal chromosome, neither of the chromosomes will have the PWS genes because they're inactive in the maternal copy as represented by the blue. So these genes that are in this region will not be expressed. So the child will have Prater-Willi syndrome. If the deletion of this region of chromosome 15 happens in the maternal chromosome, Angelman syndrome genes, which are only expressed in the maternal copy, will not be expressed, and the child will have Angelman syndrome. It's because in the paternal copy, these genes are inherited as an inactive copy. As it pertains to this chapter, the next lecture will explore other aspects of transcriptional regulation, as well as the regulation of gene expression at the levels of RNA processing, translation, and protein modification. We will also discuss some of the most important techniques that are used to study and to manipulate gene expression.